Hello everyone, welcome back to my YouTube channel. Welcome back to another video tutorial related to molecular basis of inheritance. So till date we have studied about the structure of the DNA, discovery of the DNA. In today's video lecture we are going to start about the different types of RNA, about the RNA or ribonucleic acid, what are the different types and their functions. So let's begin with the class. So RNA or ribonucleic acid, what is the chemical component of them? Just see first their structure. That is, here comes the ribose sugar. This is nothing but the ribose sugar which is differing from the DNA. In spite of not having any oxygen in the 2' carbon atom or number 2 carbon atom, it is holding oxygen. That is why it is ribose, not deoxyribose. In case of DNA, there is lacking of oxygen molecule. That is why the name of the sugar is deoxyribose. But in RNA, there is no lack of oxygen molecule. That is why the name of the sugar is ribose. And that is holding the phosphate group in one of its hand. That is in the 5' carbon atom. That is holding the phosphate residue or phosphate group. And in another hand, it is holding nitrogenous bases or N2 bases. It is holding nitrogenous bases or N2 bases. So there are basically two types of bases which are found here. One is purine and another one is pyrimidine or pyrimidine. In case of purine, there are further two types of purine components which are found here, adenine and guanine. These are same like DNA, but in case of pyrimidine, we can found uracil in spite of thymine. That means in case of DNA, there was thymine, but in case of RNA, there will be only uracil and pyrimidine will be cytosine. That means how the bond formation will take place between them. Adenine will form bond with the uracil residue by 2 hydrogen bonds or double bond, and guanine will form 3 hydrogen bond and with the cytosine. So this will be the nitrogenous base pairing, if any kind of base pairing is occurring or not. Why am I saying so? Because in most of the cases, generally the RNA is single stranded in nature, but in few cases and sometimes the RNA structure, that is the single strand, can form some loop kind of a structure, and when the two strands will come parallel to each other, then two complementary bases can form pair between them, and at that time only this kind of base pairing can occur. A can be paired with U and G can be paired with C. Guanine can be paired with cytosine. So this is the chemical makeup. So that means how we can define RNA or ribonucleic acid. Ribonucleic acid basically a chain of polynucleotide molecules. This is a chain of polynucleotide molecules. Now what is the meaning of polynucleotides? Polynucleotide means multiple number of nucleotides or more than one number of nucleotides residues are present. So what are the basic components? Sugar, base and phosphate. So sugar, base and phosphate, these are the components of nucleotide, and when this phosphate is not present, only sugar and base is present, then this component can be considered as nucleoside. So I hope that the concept between nucleoside and nucleotide is clear. When phosphate group is absent, only ribose and only sugar, and this is the sugar. So when only the sugar and base is present, then it can be considered as nucleoside, and when sugar, base and phosphate is present, then that is considered as nucleotide, and when more than one nucleotides are forming the chain-like structure, that is forming ribonucleic acid or RNA. Now let us move ahead into the different types of RNA. What are the different types of RNA which are found in the living organisms? So basically there are two types of RNA which are found in living body. One is genetic RNA and another one is non-genetic RNA. Now firstly let us see the case of genetic RNA. The question can come in your mind that I have said already that DNA is the genetic material in most of the cases. So how the RNA can be the genetic? That means what is the meaning of genetic RNA and so. So in few cases, like in most of the plant viruses and in few animal viruses like influenza, there is the lacking of DNA. There is no genetic material of DNA, saying DNA. So what will happen? There will be no such thing which can carry the information, which will be carrying the blueprint of life and which can transfer the information from one generation to other generation. That will never happen. So in that cases, RNA acts like the genetic material. Okay, so in that cases, RNA will act like the genetic material, and in such cases, the RNA are considered as genetic RNA. So where we can find genetic RNA? In most of the plant virus and in few animal viruses like influenza. So influenza is the type of animal virus where we can find the genetic RNA. And what about the non-genetic RNA? These are non-genetic, means where in most of the cases, where in most of the animals or organisms where DNA is present as the genetic material, in such cases, in such cells, the RNA is also present. Though DNA is present and representing the genetic material, but RNA is also present and performing some role. So in such cases, such RNA are considered as non-genetic RNA. Those are considered as non-genetic RNA, and there are basically three main types of non-genetic RNA which are found in the eukaryotic cells. What are those? Let us now see that. There are mainly three types of non-genetic RNA: mRNA, rRNA, and tRNA. These are the three main types of non-genetic RNA which are found in the eukaryotic cells, as well as in the prokaryotic cells also they are present. Okay. Now let us see one by one that how they are differing from each other, what is their structure, what is their actual characteristic features, and what are the functions they are actually performing. Okay, so let us now see that what are the structures and components, or what are the characteristic features of all of these, like mRNA, rRNA and tRNA. So firstly, let us talk about the mRNA. What is the meaning of mRNA? mRNA means messenger RNA. mRNA means messenger RNA. M for messenger, messenger RNA. Why the name messenger? Because this RNA, actually what it will do, it will carry all the information, all the messages, whatever is stored in the DNA from nucleus to the cytoplasm. Okay, so within the nucleus, there is the DNA who is carrying all the information for basic structures, functions and everything. So whatever information it is storing, the mRNA will copy all the information to it and it will carry it. It will convey its message from nucleus to the cytoplasm so that the protein synthesis can occur, because DNA is mostly carrying the information for protein synthesis, which protein has to be synthesized and when. So that code or that message has to be sent to cytoplasm, because ribosome is present in cytoplasm. It is not present in the nucleus. So, and we know that ribosome is the protein factory. So to make the protein, it is very, very essential to reach to the ribosome. The information has to reach to the ribosome. Other than reaching to ribosome, it is never possible to make the protein. So to ensure it, messenger RNA is formed, which is copying all the information to it from the DNA, and it will come outside through the nuclear pore complex to the cytosol or the cytoplasm, where it will start the protein synthesis mechanism. This is the main function of the mRNA. It is taking part in the protein synthesis. It is taking a very important role, but it is found very less, in a very less amount, like 3 to 5% among the whole RNA, whatever is found, like mRNA, rRNA and tRNA. Among these three types of RNA, mRNAs are found only 3 to 5% in the cell. So can you imagine that how much less these are? Why so? Because right after forming the protein, the mRNA actually gets destroyed. So these are very, very unstable also. Very unstable. So let me just write it down that this is very unstable. What is the reason behind its instability, or why it is very less stable? Because it is a single-stranded molecule. Let me just draw a picture of mRNA. How the mRNA is looking like. This is actually a single-stranded molecule like this. This is so simple. and here the alternative sugar and phosphate is present like the DNA. Like if this is the sugar, it is holding phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. In this way, this is present, this is the backbone, and all the sugar is holding the nitrogenous bases. So these are the bases. These are the nitrogenous bases which are held by the sugar. So obviously there is 5' end and 3' end also like the DNA molecule. So there will be 5' end and there will be 3' end also. So as simple as that, the structure of mRNA is as simple as that. That is why, and if sometimes they will form some loop-like structure, like if they will form this kind of structure, then also there will be no formation of base pairing among themselves. So that is why it is so simple molecule and very reactive, and that is why it gets destroyed very easily, and their number is so less, that is almost about 3 to 5% among all the RNA which are present in the eukaryotic cell. Now mRNA actually is formed inside the nucleus. How it is formed? By the process of transcription, the mRNA is formed. And as it is actually copying all the information to it, so it is forming by making any one of the strand of DNA as parental strand, it will be formed. The strand of mRNA will be formed. So we can say that all of the nitrogenous bases, whatever will be present, that will be complementary to the DNA. So let me just make it easier for you. Say for example, there are bases like A, T, G, G. So this is present in one strand of DNA. Then on the other strand of DNA, there will be the presence of T, A, C, C. This will be the other strand of DNA. Now let us say for example, by making this strand as the parent, the mRNA is forming. So what will be the nitrogenous bases which are present there? T will make its copy, its complementarity with A. So the first nitrogenous base will be A. Next nitrogenous base will not be T, because A can make its base with the T, but in case of RNA, T is absent, thymine is absent. In place of thymine, it is replaced with uracil. So that means uracil will be here. Next with C, there will be G. Next with C, there will be G. So these will be the nitrogenous bases which are present in the mRNA. And exactly this will be complementary strand to this strand. That means this strand will be similar to the another strand of DNA. Another strand of DNA which is the non-parental strand. So this will be similar to the non-parental strand of DNA. Right after formation inside the nucleus, there happen some changes. Why does changes occur? Because I have already said that this is very simple molecule and very unstable in nature. So it has prone to be attacked by some RNase enzyme, ribonuclease enzymes which are present in the cytoplasm. So right after formation of nucleus, if there will be no modification, no protection will be taken, then the mRNA will come outside in the cytoplasm, where so many ribonuclease enzymes are sitting there to digest the RNA. So it will go and they will start digesting the mRNA molecule. So it will not be functional at all. So that is why protection is very much necessary. That's why a little bit of modification has to be done there. So what is the basic structure of mRNA? Now let us move into that after doing all the modification and everything. What is the final structure or the actual structure of mRNA? Let us now move ahead into that. Okay. So in the 5' end, there happens an addition of some unusual group of molecule that is known as 7-methylguanosine. This is a very unusual group. 7-methylguanosine. So this is a very unusual component of guanine or guanosine. That is 7-methyl. Methylation in the guanine residue will happen. That will make this component insecure. This will actually ensure that there will be no chance of recognizing mRNA with the help of by the RNase or ribonuclease enzyme, and by that way the mRNA gets protection. Okay, so that is why 7-methylguanosine, this kind of capping will happen here. So this is the cap. So it will wear one cap on its head. What is the cap? 7-methylguanosine. So how the mRNA will remain protected? With the by wearing a cap which is made up of 7-methylguanosine. This is a very unusual component which cannot be recognized by the enzyme RNase or ribonuclease. So in that way the mRNA will remain protected. But it is not only necessary to protect itself from the head. It is very much essential to protect it from the tail portion also, from the another end also, from the 3' end also. That is why addition of lots of adenine molecules will happen. Lots of adenine molecules will start to be added. That is why this end is often considered as poly A tail. This part is often considered as poly A tail, and polyadenylation is happening in this 3' end. How many adenine residues can be added up to here? Almost about from 200 to 250 molecules of adenine can get attached to the 3' end. So that a long adenine residue will be there, poly A tail will be formed, and mRNA will remain protected. This is the main concept behind it. But apart from that, right after starting with the 7-methylguanosine, after that there are 50 to 100 bases, nitrogenous bases, which will not suddenly take part in the protein synthesis mechanism. Which will not suddenly take part. They are not taking part in the protein synthesis mechanism. So I am just drawing it like this so that you can understand. There are almost 50 to 100 nitrogenous bases which are not taking part in the protein synthesis mechanism, and this region is considered as non-coding region 1. This region is considered as non-coding region 1. Right after that, the bases will start. These are the coding region. Now let me tell you that what is the meaning of coding. I will tell you in much more details about the coding and everything. But for now, today's class, just let me tell you that what is the meaning of coding. Actually, what happens is the only 3 nitrogenous bases will be present there which can code for a particular amino acid. And that 3 nitrogenous bases are considered as codon or code. So that is why this region where the functional, these are functional zone where these are containing the information for the amino acids, that region is considered as the coding region. So let me write it down here. This is the coding region. This is the coding region where the functional groups will be there, or functional nitrogenous bases will be there which are carrying the information for the specific amino acids. So specific amino acids will come one after another. They will remain attached with each other with the help of peptide bonds, and they will form the protein molecule. So that is why this region is considered as coding region. So from any one place, this protein synthesis has to be started. So there is a particular start codon that is considered as AUG. Oh, surprisingly, I have made here, this is AUG. I have not made it intentionally, but I can see that I have already made. So AUG is the start codon. For now, you just keep it in your mind that AUG is the start codon for each and every organism. This is universal. Okay, this is constant and universal. So there will be AUG, which is the start codon, and there will be UAA or UAG or UGA, which are considered as stop codon. So there will be initiation zone in the coding region. There will be initiation zone and there will be stop zone also. So initiation zone, stop zone, these are the parts of the coding region. So right after the 7-methylguanosine, there will be a zone which is, there are 50 to 100 nitrogenous bases which are not at all taking part in the protein synthesis mechanism. So that region is known as non-coding region 1. After that, the coding region will be started with the AUG sequence, that is the stop codon, and after that, right after that, several other sequences will come, that is the coding region, main coding region. After that, it will be ended with the with this sequence. That is, with these codons we can say UAA, UGA or UAG, which are the stop codon. That means where the protein synthesis has to be stopped. That means right after this stop codon or stop region, say for example this is the stop region, and right after that there are much more 50 to 100 residues which are not taking part once again in the protein synthesis, and that region is considered as non-coding region 2. Non-coding region 2, and after that the poly A tail will be started. So this, and after that the 3' end, and here the mRNA residue is ended. So this is all about the structure of the mRNA. Keep this in your mind that codons are very specific to the amino acid. That means one codon, that is, say for example here I have written AUG, that can only code for methionine. There is one amino acid which is considered as, which is known as methionine. So this methionine can be coded by AUG. So in this way there are several other amino acids which can specifically code for a specific amino acid. Just randomly these cannot be chosen. Okay. So this is all about the mRNA, and in this way this is taking a very important role in the protein synthesis mechanism. Stay tuning to my channel, and if you found this video helpful, then please don't forget to like, share and subscribe to my channel, and comment down below if you have any question related to this topic. Thank you.